Good afternoon, dear participants of the conference. My name is Yulia, and I'm a student of Research and Development Center of Biomedical Photonics of Oral State University. First of all, I would like to thank the organizers of this conference to the opportunity to be here and to make a report. And today I will talk about the wearable flowmetry devices for study the effect of hyperventilation and hypoventilation respiratory exercises on the blood mucocirculation. Okay, so currently there are three types of breathing. There are upper, middle, and low. But these types involves in the breathing only a part of the lungs. And its breathing is unproductive because we spent a lot of energy on these processes, but the result is minimal. And full breathing exercises uses the whole volume of our lungs and allows volunteers to change frequency of inhale and exhale. So also these exercises actively using for rehabilitation after diseases of respiratory infects and after other diseases of all organism. So, but however, there are a lack of research of the effect of this breathing, full breathing, on the microhemodynamics. So, because of it, the aim of my talk was to study the effects of hypoventilation and hyperventilation yoga breathing exercises on the parameters of microcirculation and their relationship with parameter of spirometry and also with gas exchange. So in this study, we used the laser Doppler flowmetry method, which based on the probing of tissue and analysis the light which be reflected from red blood cells. The recorded parameter is index of microcirculation. You can see it on the slide. And this method allows us to monitoring the mechanisms of regulation of the microcirculatory system, like respiration, cardiac, and also work of endothelial cells, precapillary sphincters, and also innervation. We used in this study, we used the distributed system of 6 portable laser Doppler flowmetry analyzers. You can see it, and each analyzers have an identical channel for recording the skin blood perfusion and also movement and skin temperature. For recording the gas parameters, we use a spirometer with function of spirometry, of course, pulse oximetry, and so on. Something about our protocol. They consist of 3 stages. At first and third stage, all volunteers breathing normally. And on the second stage, they make full breathing exercises with different frequency, hyperventilation and hyperventilation regime. All volunteers were in sitting position. And areas of interest in the study was the areas of supraorbital areas, third fingers, and first toes. We studied the symmetrical areas, left and right sides. So something about the technique of full breathing. This consists of abdominal, chest, and clavicular breathing on the inhale, but the exhale performed by relax of muscles. And also at this slide, you can see the LDF grams of index of microcirculation and also the wavelet spectrum for different regime of breathing. The first and the second case were performed by a specialist in this breathing, and we can see only one peak at frequency of breathing. But the third case was made by a beginner in these exercises, and we can see 2 peaks, one on the frequency of breathing and another. We can see that breathing of this volunteer was spasmodic, and because of it we can see this picture. Then we understand that we can control the technique of this full breathing with our analyzers. And some about our results. We found them that skin perfusion was increased after the full exercises in hyperventilation regime and also after the hypoventilation regime. But the nutritive blood flow increased only after the hypoventilation regime in all study areas. So in the region of supraorbital arteries and fingers, we see a little increase in the amplitude of myogenic oscillation that was higher after the hypoventilation regime confirmed to hyperventilation regime. But in the it also it can indicate an increase in the number of functioning capillaries and a decrease in the tone of microvessels in arterial department. But in the toys, we see another situation. The oscillation of neurogenic oscillation was high in the volunteers after they performing the hyperventilation regime confirmed to the hypoventilation. Okay, so some results about gas analysis. We can see that regime 10 to 10, I mean the 10 seconds for inhale and 10 seconds for exhale. For this regime was a hyperventilation because we see a increase in the minute volume of breathing and also in the percentage of oxygen in the inhale area. And the regime 2020 and 3030 was a hyperventilation because this parameter will decrease. And then we see a relationship between microcirculation parameters and gas analysis, and we found correlation between respiratory mechanisms and percentage of oxygen, and it means the compensatory reaction of our body to the decrease in oxygen blood. So, and the correlation between the nutritive blood flow, neurogenic, myogenic, and cardiac oscillation. This oscillation means the arterial blood flow in capillaries with the minute volume breathing, and saturation. So this correlation can allow us to see consistency of respiratory system and oxygenated blood flow delivery to cells. I say something about conclusion. Only a hyperventilation mode leads to increase in the number of functioning capillaries and the cell nutrition, and which can be a result of gas exchange and some metabolic processes. This work may be useful to understanding the interaction of oxygen production and its delivery to tissues. Also our devices, portable devices for recording microcirculation parameters can be an instrumental method for control the performance of full breathing exercises and their effectiveness in rehabilitation on the microvessels level. And in the end of my talk, I would like to express my gratitude to the volunteers who took part in this study and to my co-authors. Something about our geography, and that's all about my talk. Thank you for your attention. I will be glad to answer your questions.